Skip's interests are eclectic. His impact has been significant in many areas of related science to this workshop, but in particular, his work on the role of chronic viral infection, persistence, manipulation of host immunity by chronic viral infection, and the whole role of viruses and non-traditional mechanisms for chronic disease of uncertain etiology. So, Skip, thank you for being here.
That means you've actually looked at these things and understood what I was talking about. Thank you for that introduction.
No, the first thing about the second.
Thank you. I'd like to thank the organizers for the opportunity to speak. This talk is going to have a couple of things that have come up in questions, that is viruses and RNA. Okay? And I'd like to request your willing suspension of disbelief in the following way. Think of what's happened in bacterial microbiome studies. From the beginning of the technologies to be able to quantitatively assess a broad range of bacteria that you can't easily detect by standard methods over the last about decade or so, there's been this incredible blossoming of studies of how bacteria are related to lots of different things. And so I want you to suspend belief that bacteria are the only things that are important for the next half an hour. Okay? So I'd like to first acknowledge the people in my lab who do a lot of the work. So we study the role of specific genes in inflammatory bowel disease. You'll see me come back to that. Most of today's talk is using metagenomic approaches for viruses in either animals or humans or primates, which are related to diseases. We're very interested in chronic infections, and a piece of the lab is involved in defining the molecular mechanisms responsible for persistence. And I'm going to use some of those tools in this talk, mutant viruses and such, but I shan't be talking about that in detail. And then when you do viral pathogenesis, it takes a village, as Hillary Clinton has said to have said. There are a lot of different expertises that are required. And so we have a number of different people who have collaborated with us, Romnick that you'll hear about, Jeff Gordon at the end in particular. So what I'm going to talk about today is I'm going to have three concepts and then data related to them. So the first concept I want to briefly introduce is this idea that there's a virome. It's a key part of our metagenome. It's on the inside of us and on the outside of us. Then I'm going to show you some data which is related to the idea that a persistent herpes virus infection is symbiotic for us, and it controls our innate immune system and basal gene expression in tissues so that a chronic virus might alter who we are and how we respond to our environment. And I'm going to introduce the concept thereby that the virome may influence the immunophenotype of the host. And I'm going to show you some data about a different kind of persistent virus which triggers a specific inflammatory bowel disease phenotype only when a certain gene is mutant in the host. Now, this is a genetic argument. That means that virus plus gene defines phenotype, so that you can't define the phenotype of the host just from the genotype. Okay? And then, arguing that the virome is important, I'm going to tell you two brief stories about the scope and nature of this enteric virome. And through this talk, realize that it is the technologies which allow us to sequence everything, DNA plus RNA, and then analyze it computationally that allows us to expand out from looking at bacteria into looking at viruses, and now we're looking across other taxa. So the chronic viruses, thank you, Gerard, for introducing this. Everyone is familiar with this. I know everyone in this room loves this. But this is what's inside you. So all of us are chronically infected with multiple viruses. And in fact, if you total them all up, if this is the number of people on Earth, this is the number of people infected with each of these viruses. On average, there are 10 per person. Each of you has about that many. Some of them are making 10 to the 12th particles in your circulation per day right now. They are all under immune surveillance. This is not a static interaction. You are constantly containing these things. You can't get rid of them. Your immune system is not good enough to get rid of them, and they are in balance with your immune system. many of them are performing that particular balancing act in your mucosal tissues, the places where then you interact with bacteria or other invasive species. So this is who we are, in a dynamic equilibrium with a lot of viruses on the inside and some on the outside as well. We think this is a very important concept, and it is identical conceptually to the idea of the bacterial microbiome as a set of organisms which lives on or in us and then defines our physiology in important ways. So that's the concept. So now I'm going to talk to you about some of the experiments that got us there. So we're interested in herpes viruses, and about a number of those persistent viruses that you all have, on average you have 4 or 5 of them, are herpes viruses. And we have come to believe that they have co-evolved with us for a long time, and that confers both advantages and disadvantages for the host. How long have they evolved with us? The ancestral herpes virus genome is shared between birds, reptiles, and mammals. So every single species, including us, has had our own herpes viruses from the time we speciated into separate species. So these things have been studying us, and we have been studying them evolutionarily for a very, very long period of time, probably in the range of 100 million years or so. I'm going to talk to you about an advantage to the host, which is that these viruses are not silent inside of us, but rather they activate our innate immune system and can thereby protect us against invading bacteria. And then they have disadvantages for the host, and I'm going to set aside the boring standard way in which viruses cause disease, which is that they go into a cell and they kill the cell, and that makes the liver sick if the liver cells are dead. Now I'm going to talk to you about other kinds of mechanisms by which a virus might cause disease. In this particular way, an interaction with a host gene. So this is a very simple experiment. I'm going to go through this quickly. This was published in 2007. We studied latent herpes virus in mice. We've made a virus, by the way, that can undergo acute infection but can't persistently infect the host. I'll come back to that virus mutant. But we did a simple experiment. We infected mice with this virus, or mock-infected, and then we challenged them with a very high dose of Listeria monocytogenes, which would kill the uninfected animals, and these animals lived. So latent infection prevents you from dying. 1 month after infection, 3 months after infection, lasts at least 6 months, which in the lifetime of a mouse is a substantial period of time. What is the mechanism of this? We developed a hypothesis that this latency-associated protection is due to latency setting the level of innate immunity. And I'm just going to go through the things that we showed in support of that hypothesis. So Listeria is controlled by activated macrophages. So we asked in the latently infected animals, are the macrophages activated? Yes. Is it general to more than one latent virus? Yes. We tried it with a mouse CMV, which is related to human CMV, HHV6 and HHV7 of humans. Does it require latency or just infection? It requires latency because if you infect with a mutant virus that we made that can undergo acute infection but can't undergo chronic infection, there's no protection. Is it innate immunity? Well, it is, because you don't need effector T cells to be present at the time of the Listeria challenge. It's an innate response that makes you resistant to the bacteria. And of course, if it's innate immunity, it shouldn't be antigen specific. And it's not. It protects against both Listeria and the causative agent of plague, Yersinia pestis. Okay? So that suggested to us that macrophage-based innate immunity is controlled by latent infection. Remembering again that we all have multiple latent infections. So we thought about other parts of the innate immune system. So if you were a natural killer cell biologist, and you were a human natural killer cell biologist, you would tell me as a mouse person that I study irrelevant cells, because the mouse natural killer cells are fundamentally different than human natural killer cells, because human natural killer cells, if I bled all of you, would express the lytic proteins that kill infected cells called perforins and granzymes, but mouse natural killer cells do not. So that's a genetic difference. Let's not study mice. So what we did was we latently infected animals, and we asked whether the latently infected animals' natural killer cells express granzymes, which are the thing that distinguish mouse from human. And we humanized the mouse by latently infecting the mouse. But if we put in a virus that can't establish chronic infection, it doesn't do that. It suggests that the mouse is not genetically distinct from humans. The mouse is environmentally distinct from humans. And secondarily, that another part of the innate immune system, being natural killer cells, is controlled by latent infection. So then we thought, well, is that functionally important? So if you take animals and challenge them with a natural killer cell-dependent tumor, they die. But if they're latently infected, they do not. So these latently infected animals have activated natural killer cells that protect them from a cancer. And that's natural killer cells because if you deplete the natural killer cells, these animals now die of the cancer. So that suggests that the fundamental nature of the immune response is determined by this latent virus, maybe other parts of the host virome. How about other stuff in the host? So we did a very simple experiment, which is now unpublished, writing it up now. We latently infected an animal, we pulled out the organisms, and we did microarrays to look at gene expression. Maybe lots of genes are different when you're latently infected. So this is the wild-type virus. This is the mutant virus that does not establish latent infection. This is liver, spleen, and brain. There's a whole set of genes which are changed in the spleen during latent infection. If you now look, this is the spleen set. This is a set that's changed in liver, and a different set are changed in brain. So that the basic nature of the transcription in the organs of an animal is defined by whether the animal is latently infected with a herpes virus. Of interest, these are a series of genes which predispose or are in loci which are linked to predisposition to human diseases, celiac disease, Crohn's, multiple sclerosis, and a number of these genes are changed in the latently infected animal, suggesting but not proving that your susceptibility to disease might also be altered by your endogenous virome. So this gives rise to the concept that if there are advantages and disadvantages, perhaps there's an immunophenotype, at least in the innate immune system, which is the individual's normal immune responsiveness, and that's some summation of genetic, developmental, and environmental factors. This is not a new concept, but I'm saying that the viruses are one of those environmental factors. So the virome might then contribute to immunity, inflammation, and disease by influencing this phenotype of our host, of our cell, and therefore the virome, and we already know the microbiome, you've seen that repeatedly, maybe the fungome, the phagome, the parasitome, all alters this, and that defines the field upon which pathogens or invasive species play their roles in causing disease. So, are there other ways that you can have an interaction between a chronic virus and a host that might predispose to disease? And this is a story that is from a different virus, a norovirus, which I'll introduce in a little bit. It'll take me a few slides to get there. Noroviruses cause 95% of the epidemic non-bacterial gastroenteritis in the world. The human viruses have never been cultured. And a proportion of you are shedding noroviruses even as we speak. They're completely human-specific, but they cause epidemics. And that's because some of you are shedding them, and when you get it into the mayonnaise at the church picnic, you give it to the other people who don't have it, and you get an epidemic of severe gastroenteritis. Okay? So they're persistent and acute viruses. So this story starts with, we have been studying autophagy as a host defense mechanism, and after we'd made some mice, which initially had no phenotype, very depressing, these papers came out linking this autophagy gene to Crohn's disease, multiple papers. And so we looked in the mouse, and I'll tell you the story of what we found. But first I have to tell you a little bit about the small intestine. You saw some beautiful pictures of the small intestine in our talk on hibernation earlier. There are these villi, and then there are crypts. The bacteria live in the lumen, and the crypts have this wonderful cell in them called a panicell. A panicell is an epithelial cell which is responsible for managing the crowd. It makes granule contents, granules, and then releases them. And these contain lysozyme and some of those antimicrobial peptides you've heard about, and they regulate the ecology. So there's this symbiotic feedback process here where the panicels are regulating the microbes, and this is part of your normal physiology. So we have two components here, an autophagy gene and a panicell. So because the human gene was linked to Crohn's disease, we decided to look in the intestine. And we took wild-type mice, and we made mutant mice that were hypomorphic for expression of the gene. And we just did a simple experiment, which we stained the mucus, which overlies the villi, for lysozyme, which is in these granules. And if you could turn down the lights, that would be great. What you can see here is this is mucus from the gut, and there's no red in it, and this is The red lysozyme coming from the granules, that's a wild-type animal. That's the mutant. So the mutant animal has not got granule contents out in the mucus. So we decided to stain the Panna cells where this comes from. This is the base of the crypt. The blue here is the nuclei, and there's these beautiful granules. Okay? This is normal, and this is what the Panna cells look like from these animals. The granule exocytosis pathway is massively disrupted, and we thought that this disorganization was a reasonable explanation for why there's no secretion. Now, and one of the most fun things that happened in this project, we had a group of collaborators who've been harvesting human samples from resections from Crohn's disease, and that allowed us to do the following experiment, which is the identical experiment to what I just showed you, except now we're going to take patients with Crohn's disease who are wild type or who are mutant for the ATG16 risk allele. This is human crypt, nice normal Panna cells with the granules. 100% of the humans with this homozygous mutation have abnormal granules. You can see this very pretty granules here in the immunofluorescence, and you have these very abnormal cells in the humans. So think for a moment what we just did. We took a human risk gene, made a mutant mouse, looked at the pathology in the mutant mouse, and found that humans with mutations in that same gene phenocopy what we found in the mouse. Okay, so that's pretty cool. We thought that was great. Okay. Now, we like to study immunity to viruses, and so we created a mouse facility, which we took these abnormal Panna cells in these mice, and we rederived it into a facility where everything in the facility is autoclaved except the people and the mice. Okay? And this is not germ-free, but this is very, very clean. No postdocs, no graduate students. I cannot walk into it. Okay, you have to pass a test from the people who are in it to be able to walk into this facility. And that is, we've rederived hundreds of mouse strains into this. I'll just tell you parenthetically that all you people who study autoimmune models, when you do this, the phenotype shift. Okay, I presume some of that's the microbiome, and some of that's probably the virome. And what we did is we did this, and we normalized the animals. Now remember, we phenocopied humans with this, so we were excited about it, but when we rederived, now the Panna cells are normal. Okay, so there must be some environmental factor. Could stress, maybe an infection, trigger that? Well, now part of our lab does pathogen discovery, and we had the great pleasure in 2003 of finding the first murine norovirus and solving the problem of culturing noroviruses for the first time as well. And that has allowed us to have all the fun that a virologist can have in public. Okay, by sequencing it, finding the structure of it, making reverse genetics, mapping the genes that are involved in various and sundry things. And Larissa Thackeray did a very nice experiment, found 32 different strains from around the world of this virus, and she found that some, thoughtfully colored blue, can infect acutely but are cleared and are not persistent. But others, which will be coded in this color, this is structure that we did with Tom Smith, they persist. So we have non-persistent and persistent viruses. Everything I'm going to tell you requires a persistent virus, and this virus doesn't do it. Okay? So now, we took this, and we normalized it, so it's not developmental, it must be environmental. We gave this virus, and 100% of the animals have this phenotype. So we took a human phenotype mimic, cleaned it up, and reinduced the human phenotype mimic by giving a virus. Now, this is not due to a direct infection of these Panna cells. This is due, we think, to infection of dendritic cells, which then release a factor which tells these cells to do something which they don't do very well when they have a mutation in this autophagy gene. So this is not a standard pathogenesis model. So if you now take an experiment where you take the mutant animals, give the virus murine norovirus, and then you challenge with a chemical agent that damages the intestine, you get new phenotypes. So you raise the animals clean, they have normal Panna cells, you give them DSS, they have a little bit of colitis, and if you look in their small intestinal villi are normal. Now you take these same animals and give them the persistent virus, but if I put the blue virus here, this wouldn't happen. The non-persistent virus doesn't do it. Panna cells are abnormal. Now we get a completely new phenotype, transmural inflammation, lymphoid aggregates. This has some aspects that are similar to Crohn's disease, which was the risk gene came from Crohn's disease. And now we get a completely new phenotype, which is allele atrophy, which was not present here. So what I'm telling you is that the virus plus the gene is responsible for new phenotypes. And from a genetic point of view, therefore, you cannot merely interpret the phenotype of the gene without its trigger. So we did an experiment which I think is probably one of the more interesting ones, and we laser captured the cells which were either triggered or not. And we did gene expression profiling, and this is now protein targeting, metabolism, carbohydrate metabolism, protein trafficking. And what you can see is that the response of the virus in the mutant animals is not qualitatively different from the wild type. It is flipped. That is that all the genes which are going up are going down for these genes. Think about what that means. What that means is that when you look at the variation between individuals, the presence of a virus with a certain allele could give you the completely reversed transcriptional response to a stress. What about the mechanism of this? This is virus triggering of increase in muscle width, but a number of other measures are appropriate there, and you get the same thing. So this is the virus triggered in the red here. In humans, you can treat Crohn's disease with anti-TNF. That decreases the viral trigger. Interferon gamma starts to play a role. Anti-interferon gamma makes the virus trigger go away. And then in my favorite experiment, which the postdoc, who's a brilliant guy, who's now a junior faculty member at NYU, Ken Cadwell, initially refused to do, which I will never let him forget, we decided that we would test whether bacteria were important for this virus-triggered disease. So we gave antibiotics and cured the virus-triggered disease. So that means that when you trigger a disease in the setting of a specific allele of the host, you cannot interpret the response to antibiotics as indicative that the bacteria were the sole cause. So the microbiome can interact with these viruses in order to generate novel phenotypes. You already knew this because in the 1918 flu pandemic, people died from bacterial pneumonia. And if you didn't know anything about flu, you'd have thought that people were dying from a bacteria, but it was really the flu predisposing to the bacteria. So this is a standard thought. Okay, but, and one other just little fun thing. So it turns out, remember I told you the blue virus doesn't do this? They're the same serotype. Remember, you probably mostly think about viruses being distinguished in humans because they're serotype difference. Flu A, flu B, et cetera, et cetera. The virus that triggers and the virus that doesn't trigger are both the identical serotype, which indicates that you can't use a serotype response to a virus to rule in or out that the virus triggered your disease. So the concept here is that the virus does something to the host, and that would have a normal viral phenotype. And if you had a complex pathway like autophagy, by which cytoplasm is delivered to lysosomes to degrade things, and ATG16 is just one gene in that pathway right there, you might have a gene phenotype as well, but when you put the virus and the gene together, you get a new phenotype. So that means that you should not be solely considering these inflammatory diseases to be genetic alone or microbe alone. You all know this already because you would have put the microbiome here. I'm just putting a virus here. Okay, so that's the concept that the virus can interact with the gene. Think then of the diversity of viruses and the diversity of allelic distinctions between an outbred species, such as humans. So what that would mean is that if there was a large diversity of viruses and a large diversity in the allelism of the population, you could get a number of different outcomes related to disease. Remember also that in most estimates of high-throughput GWAS-type studies, the proportion of total risk which is conferred by the observable genotypic differences is somewhat less than 20%, even within studies which have, as for IBD now, 163 identified loci, 600 odd genes in those loci from 75,000 patients, not work from us, but from others, and 25 odd thousand controls. Those are so in that you're still less than 20%. So there's a lot of variation in phenotype, which is as yet unexplained. Okay? Rick and I are nodding at one another over here because the IBD field is really cool, okay? There's a lot of fun stuff going on. So that's the concept. So how big is the virome? And this is where you learn that you should sequence RNA and DNA. Okay? So I'm going to tell you a story in primates with AIDS, infected with SIV, take their stools, filter, take total nucleic acid, convert the RNA to DNA, sequence everything by shotgun sequencing. We've done this by Illumina now. And then we developed a software package which most of the things that people do are targeted to mapping a read very specifically to an already existing genome. This is to look for things that are very distantly related, okay, so that you can find new viruses. And then we can look at the taxonomy using Megan. And the idea here is that there is an association between SIV AIDS and HIV AIDS with abnormalities in the gut, which is translocation of what's thought to be the microbiome across the gut to stimulate systemic immune responsiveness. So the idea is that there's something wrong with the gut and that you transfer bacterial PAMPs and LPS and other things into the circulation that activates the immune system, stimulates the replication of SIV and HIV, and actually that translocation is argued to be a better predictor of progression to AIDS than CD4 count or RNA level. There's something about the gut. Now currently, before this work, it was thought that that's a Th17, which we heard about earlier. Difference between HIV and SIV is changing your immune responses to the bacteria in the gut, and somehow that changes the epithelial permeability, and you get leakage across into the systemic population, circulation, and that activates your immune system. And that is no doubt correct. But it's not necessarily true that that's the only thing that could be damaging the intestine. In fact, if I had AIDS and I had a lesion in my head and you were going to treat me, you would not presume that the lesion in my head was due to an abnormal Th17 response. You would think I was immunocompromised and you would consider that I had a pathogen. The gut's about the only organ in AIDS where people don't think that way. So we took this approach, and then we took four different cohorts, two cohorts at the New England Regional Primate Center, which had been intrarectally infected with SIV, and they were controls. And we sampled them 24 weeks after SIV infection, or 64 weeks, at which point many had died of AIDS. We had a similar cohort in Tulane, and then we had two cohorts which were infected with SIV, but these monkeys are infected with an SIV that is not pathogenic. It does not cause AIDS. They can live for years with this SIV. They, interestingly, do not have translocation of intestinal contents into their circulation. We took these animals, we did a cross-sectional study of stool, we got lots of different sequences, many of which were unique, and we asked, what did we see in this shotgun experiment? So this is the animals which did not have pathogenic SIV infection. These are African green monkeys. Blue is bacterial sequences. Purple is everything else. There's a lot of fungi and stuff in here that we just haven't even studied. Green is phage, okay? And red is virus. You can see one animal had some virus sequences, but there was no statistically significant association between SIV, which are these individual animals, SIV positive, SIV negative. Each animal on these graphs is one primate. So this is an enormous study, and we had a lot of help from a lot of people to do it. But there's no viruses here. Okay, this is pathogenic SIV infection. SIV positive, SIV negative, 24 weeks, 64 weeks. These animals died of AIDS, where the asterisks are. You can see a massive signature of viruses. Okay? Now, you can also see that animals which didn't have viruses early have viruses late. Okay? So I'm going to talk to you about what these are. And this same thing is happening. These are the SIV infected in Tulane. So this is not from one animal facility in Boston. What you can do is you can assemble all of those. It won't take longer than 5 minutes. You can assemble all those viruses, and you can align them to their most closely related virus friend in the database, and you can find that there are multiple caliciviruses, for example. In fact, we found 32 completely novel viruses that had not been previously described, and that did not count the anelloviruses or another group of viruses, which there were too many of them for us to even begin to count in the study. These are just the ones which are related to known pathogens in humans, primates, or farm animals. So there are 3 new caliciviruses. Knowing this, we could develop a PCR assay for all the 32 novel viruses. We could look and say, what are these things? We could look by the virus hunter analysis and assign these sequences to adenovirus, calicivirus, Serco, papilloma, et cetera, and then we could design the PCR test, and there was a 90-odd% correlation. That is, when we called it by the metagenomics, the PCR confirmed it, and 100% of the time when we missed it by the metagenomics, the PCR did not confirm it. So that means that these are actually present in the original samples. And then we took, remember there was a group of those animals that died of AIDS. We took those and looked at their intestines for pathology, and we found that some of them had enteritis, and that enteritis stained with antibody to adenovirus capsid, and those were the animals which had high numbers of adenovirus reads in their stool. So this predicted pathology that was not expected at the time of autopsy. We additionally found parvoviruses in the stool and the blood of animals. And we sequenced the blood and the stool organisms, and they were the same organisms. So we had invasion of parvoviruses from the intestine into the circulation. We looked at bacteria. These are family richness plots. 4 of the cohorts were adequate. We got pretty much the same distribution to what you do if you use 16S in the same animals. And we looked at every measure that we could and that the reviewers asked us to. And we found no association between bacteria and SIV by any measure, so it appeared to be virus. Now, this is in the fecal stream, not the mucosa. And so the idea here is that in SIV, there's a huge expansion of the enteric virome. Some cause disease, some translocate into the blood, and maybe that expanded virome that was not expected until you sequence total RNA and DNA would contribute to a number of things in AIDS. Barrier breakdown, movement of bacterial PAMPs into the tissues, systemic inflammation. And these are things we're studying in humans and in prospective cohorts of primates now. I hope I've convinced you that you should look for RNA. There really are RNA organisms. I suspect they're in other places, not just the gut. Persistent viruses, such as these, can be important for our physiology by changing the nature of the immune response, as does the microbiome, and by triggering phenotypes associated with allelic polymorphisms in the host. This role of the virome may be to change the bacterial microbiome. We know that is true at least in our IBD challenge model, and that's something we're interested in. Genome does not equal phenome unless the virome is accounted for. And I would argue that the lack of quantitative definition of the virome, like bacteria used to be, could contribute to our non-genetic phenotypic variation. And with that, I'll close and be happy to take any questions. If there's time for any questions.
Thanks, Skip. There are a ton of questions I think might be asked. I'm not sure whether we can get into any right this moment unless there are just questions again for clarification. I just want to make sure we have time to get back to have a more detailed discussion of some of these issues. And we are going to return to your favorite domain of life with the next speaker as well. So I guess life is an interesting term that perhaps we should talk about as well.
They're alive.
For the time being, we're going to try to preserve human function and allow a break to move around. And we'll return, let's make it 20 minutes, 5 after 3. See you then.